one of the important things about what makes a protein more likely to be broken down. For example, certain enzymes are broken down pretty quickly. They're no longer needed, so they're degraded pretty quickly. So there are specific and not so specific signals that that really result in protein degradation. The first non-specific signal is here, oxidation. You know, if a protein is damaged by oxidation or other modifications, there are cellular mechanisms to kind of get rid of it and remove it from the cell. However, there are two more specific mechanisms that destine protein for degradation, faster degradation, if you want. The first one has to do with the primary sequence of a specific polypeptide. So polypeptides that are rich in specific amino acids, or abbreviated PEST sequences, and these PEST sequences are proline, glutamate, serine, threonine. So certain polypeptides that are rich in these sequences are more likely to get degraded than other polypeptides. So that's a function of the primary structure of that polypeptide. The third important degradative pathway of proteins is really very specific and very important. It's called ubiquitination. It's basically tagging a protein, flagging it, so to speak, so it will be destined for degradation. Ubiquitin is a small protein itself. Once the cell no longer needs an enzyme, there's a process that attaches this ubiquitin molecule, or many of them, to that enzyme, let's say. Once that ubiquitin is attached, it destines that protein to go to an organelle called the proteasome, where it will be degraded by proteases inside the proteasome. So this is kind of a quick representation. So here's a protein. This is ubiquitin. You can add one or more ubiquitin molecule. That labels that protein as we no longer need it. So it is sent to this organelle called proteasome, where it is broken down into peptides. Many enzymes are gotten rid of. Some other cellular proteins no longer needed. So this is a very specific way for protein degradation called ubiquitination, or adding this tag of ubiquitin for protein degradation. Okay, you don't need to know the specific details here, but just know that when you add ubiquitin, you send it to the proteasome and you degrade the protein. If you like this video, please subscribe. Thank you.